Hi, this is chapter on cell communication, and in this chapter we're going to talk about how cells talk to each other. So what are the different ways that cells send signals in order to communicate? So there are a couple different types of things I want you to know about signaling. There are many different types of signals that cells receive. So some of these signals could be in electrical form, and if you're talking about cells such as neurons or brain cells or cells in the nervous system, these cells are going to send electrical signals to each other. So neurons do that, and the signal is called a neurotransmitter that these neurons send to each other. Another type of way that cells signal to each other are cell-to-cell contact. So sometimes cells have to be right next to each other to touch each other in order to somehow communicate. And some cells can signal really far away, and this is known as the endocrine system, where a cell can release the signal far away and then through the bloodstream sends that signal to another type of cell. So what are these signals that cells are sending and receiving? So some signals are chemical, as said here, electrical signal. Some are membrane bound, so they're stuck to the membrane, so the cells have to be close together. Some signals that cells receive come from outside of the cell, or of the body too, such as light signals, sound signals, the vibrations, as well as pressure and temperature signals. And all of these are messages that cells receive, and this is how they can communicate with their environment. So there are a few steps in cell signaling that I want you to know about. And these are basically how the cell takes in a signal and what it does with that signal in order to respond. So in general, why do cells need to communicate? Is so that they can maintain homeostasis with their environment. So once the signal comes to the cell, it has to do something with it to respond to the environment. So what are those steps? So there are 4 steps that I want you to know about. One is the signal transmission. And signal transmission is the signal traveling towards your cell. So let's say that this is the cell right here. Your signal on the way to the cell, that is cell signal transmission. The second step is reception. And reception is when the cell actually makes contact with that signal. So once it receives the signal, that would be your reception. So on your cell membrane, you have what's known as receptors, and these are proteins that can bind the signal when they come to the cell in many cases. The third step is signal transduction. So after you have the signal bound to the receptor many times don't go into the cell to tell the cell what to do. A lot of times what happens is that the receptor will talk to another molecule or protein, and that will talk to another one, and so forth until the message gets passed along. And that process is known as transduction. So passing the message along from the signal originally is known as signal transduction. And finally, the last step of cell signaling is a response. So let's say your cell needs to take more water in because it's losing water. Opening channels or opening membrane proteins, which we'll talk about today, some examples, or you talked about that last chapter. These are ways that water can get into the cell. So that would be a response to a signal that told the cell you need more water. Okay, so the response is what the cell does in response to the signal that it got. Okay, so these are the 4 steps you need to understand. And for the response, whatever protein is the one that goes ahead and allows a response to happen, that protein is called, or that molecule is called the effector. That is important there. So there are a couple of things I want you to know as well once it comes to receptors. So receptors are really important in cell signaling, and there are 3 classes of receptors that you should learn about. And these are just have a different way that they operate, but they have the same kind of idea. They receive a signal and pass it along as part of the cell signaling process. So they're the ones that work right here in step number 2, reception. So the first type of receptor I want to talk about is the ion channel linked receptor. And ions are elements that have a charge on them, okay, or molecules with a charge. So a lot of times these are sodium ions or potassium or chloride ions. And the channel, when it's open, allows the ion to go through. So these little dots are my ions. But when the channel is closed, there is nothing passing through, okay? So how this channel acts as a receptor is that it has a binding site right there, that little half circle that will bind the signal. So let's say that's the signal that came from somewhere through transmission. And once it's bound to the channel at the receptor here, it's going to open up. And once it's open, now your ions can diffuse through to the other side. So the ion channel receptor, you have a channel. It allows ions through, but it only allows them through when you have the receptor bound to the signal. The next one is the G protein linked receptor. This one, you have a protein that kind of weaves through the membrane like this. This is the receptor part. So it will bind this little signal molecule here to this spot right there that looks like a triangle. And then it's linked to a G protein. So this is the G protein right here. And the G protein has 3 parts to it, or 3 subunits. The alpha, beta, and gamma subunits. And so these subunits are able to work in a certain way once activated, which we'll talk about. And so down here, this is when you have the receptor bound to the signal molecule, the little red triangle here, or signal molecule. And once it's bound, you could see some changes that took place. So 2 of these subunits, usually the beta and gamma subunits, they come over here and bind to the receptor. That's when it's activated. And then you have the alpha subunit that goes and interacts with some other protein like an enzyme. And once this alpha subunit interacts with the enzyme, now that enzyme becomes activated. And once the enzyme is activated, that could go and activate something else. And so what I'm describing here, this activation and this activates something else, now we're talking about the transduction part of the signaling. And so this is a G protein linked receptor, because this receptor works along with the G protein in order to activate other enzymes and other proteins. Okay, and then the last one I want to talk about for these different types of receptors are the enzyme linked receptors. And the enzyme linked receptor, the receptor itself is an enzyme. And the example you have in your book is the receptor tyrosine kinase. And kinases are enzymes that are add phosphate groups to proteins. And so this type of receptor has 2 parts to it called monomers. And when they come together over here, when they're activated, they form what's called a dimer. And it's the dimerized or together form is when it's activated. And so here are the signal molecules. When they bind to the receptor, the receptor comes together, it dimerizes or forms a double connection there. And what's happening here is you have some phosphate groups being added. So that automatically happens once the 2 subunits inside the cell comes together. And that's where you have your tyrosines. And tyrosines are the types of amino acids on that protein that gets the phosphate groups added to them. And now, once this entire molecule and protein is activated with the phosphates, now it becomes an active enzyme that can phosphorylate other proteins as well. So now this is active, it will phosphorylate other proteins and activate them as well. So that is the activation process. Of course, there is a shut-off process. So kinases are enzymes that add phosphate, or is important for phosphorylation. But in order to remove the phosphate group, you would need phosphatase. So phosphatase is the enzyme that removes the phosphate groups from your proteins. And so once you remove the phosphate groups, that's one way to deactivate a protein. Or if the receptor signal and molecule now just goes away from any of these, if it's not binding anymore, it will be shut off. And so this process of going between an active and an inactive state is known as a molecular switch. And so this occurs very often in cell signaling. You have a signal that activates something, but then you have a way to turn it off. So signals are not always just turned on all the time or turned off. They go between on and off states. And kinases and phosphatases go between adding phosphate group and removing them. And again, this is what you would call a molecular switch, because it's a way to turn on and turn off a protein or structure molecularly at the molecular level. All right. So a couple other things to mention here. One is that depending on how responsive your cell needs to be, determines most times how many receptors it will have. So let's say that you only have 5 signal molecules in your cell's environment. It only needs to have a couple receptors to respond to those few signals. But let's say that suddenly a lot more signals come into your environment. Your cell won't realize that there are that many signals unless it has more receptors added to the membrane. So in order to get a big response from the cell has to now put more receptors into the membrane. And so the more receptors in the membrane, the bigger the response to all those signals. And that is what's called receptor upregulation. When you have more signals, lots of signals coming in your environment, that can tell your cell, hey, we're not responding quickly enough. Please put more receptors in the membrane of the cell so that we can respond appropriately. Now, let's say you have, all of these hundreds of signals, molecules for these receptors, and then suddenly the signal dies down and now you only have 2 or 3, not much at all. There's no reason for your cell to have 10, 20, 30 receptors just hanging out in the membrane with no signals. And so sometimes what your cell will do is do what's known as receptor downregulation, which is pretty much to take those receptors out of the membrane because there's not much need to respond to the environment at that time. So your cells are very dynamic. They go between adding lots of receptors and taking them out depending on the need for signal interaction with its environment. Okay. All right. And so one thing I didn't mention so far is that some of the receptors can actually be inside the cell. So for the most part, what we're talking about here are receptors on the cell membrane, on the plasma membrane. But there are also receptors inside of your cell. And these receptors are oftentimes what we talk about is your DNA and your genes. So certain genes can get activated and they could be not activated or suppressed or repressed or turned off. And you need some sort of signal for that to happen. And so some signals can go through the membrane of the cell, go into the nucleus, go to the DNA, find the specific gene, and activate it or turn it off. And that those types of signals are called transcription factors because they affect the process of transcription, which is the process of copying specific genes into RNA molecules that the cell will use for specific purposes. And so these transcription factors, they're signal molecules that will either activate or repress specific genes in your cell. So they're sending the signals of which genes should be active or not.